Once the 27 grams of silica gel is massed out, it's then added to a perfectly clean reagent bottle. This is a little tricky, but as you'll see in a minute, it's necessary in order to create the slurry. There we go, all 27 grams into the reagent bottle. The next step in the process is to add 70 milliliters of acetone to the silica gel. Once it's added, a cap is put in place and the mixture is mixed thoroughly. This creates what's known as a slurry, and this slurry will be used to create our TLC plates. What happens next is the slurry, after thoroughly mixing it, is added to a small plastic vial. In this case, we're using a fruit fly vial. And then some slides which have previously and meticulously been cleaned are then dipped into the slurry using a forceps. This dipping creates a film on the slide on both sides. And this is the thin layer that we'll be using in our separation process. This is usually done quickly because the silica gel has a tendency to settle in the acetone. So each slide is dipped. Oops, this requires a little bit of eye-hand coordination. And there we go. Once the silica gel is dried, the back side of each slide is carefully scraped with a razor blade so as to remove the excess silica gel from that surface. What we have now is a complete TLC plate. Mr. Hand will now make some reference marks on the plate 1 centimeter from the bottom. These marks will serve as a guide later on when we apply the amino acids to the plate. Nice job, Mr. Hand. What Mr. Hand is doing now, using a capillary tube, he's adding a very small amount of each of the known amino acids to the TLC plates. The key is to add a very small amount of amino acid to create a very small spot. You wouldn't want the spots to overlap. These spots are now allowed to dry, and then the process is repeated 5 more times to create a concentrated spot. After the final spot of amino acid is added to the plate, and while it's drying, it's time to fill the chromatography chamber with some solvent. So a pipette tip is added to a mechanical pipette. This mechanical pipette is calibrated to 1 milliliter. Pressing the plunger down to the first stop, the pipette tip is then inserted into the solvent container, and then the 1 ml of solvent is slowly withdrawn and then delivered to the Copeland jar, which we'll be using as our chromatography chamber. Very slow movements of the plunger yield the best results. We're adding 2 milliliters of solvent to the Copeland jar. This seems to be just the right amount to run our chromatograms. Covers are quickly replaced since the solvent is very volatile. It's now time to run our chromatogram. The Copeland jar is opened and our dried plate with the amino acids placed on it is now added carefully into the Copeland jar such that the bottom of the plate is just contacting the solvent. The top is quickly replaced so as to saturate the atmosphere with solvent fumes. This gives the best results. As the solvent slowly moves up the thin layer of silica gel, it's easy to see the wet portion right here of the plate and the dry portion which hasn't seen the solvent yet. This area between the two is called the solvent front. Once the solvent has reached about 1 centimeter from the top of the silica gel, the chromatogram is done. It's removed from the Copeland jar and the solvent front is permanently marked using a dissecting needle. This will aid in our measurements later on. Since amino acids have no color, they need to be sprayed with ninhydrin. This is an airbrush. It's the tool we'll use to spray ninhydrin on the plates. Ninhydrin is carefully added to the glass container of the airbrush. I'm being careful not to get any of this ninhydrin on my skin, since there are amino acids there as well. Once it's filled, we're ready to spray. Once the finished chromatogram is dry, it's placed under a fume hood, and an airbrush, which has now been attached to a pressurized tank of air, is used as a spray gun to gently spray a mist of ninhydrin all over the plate. In about 5 minutes, the ninhydrin will cause the amino acids to turn color and they'll be visible. The last step in the procedure is to transfer the chromatogram to your lab report. This is done by applying a small piece of tape to the surface of the silica gel. Rubbing your finger over the tape allows the surface of the silica gel to transfer to the tape. The tape is then gently lifted and then stuck to the lab report in the appropriate place.